Welcome back to the final part of the special senses where we wrap up hearing and we talk a little bit about balance, not too much on the balance side or equilibrium, but we talk mainly about hearing in this part four, I guess, part four. So let's talk about the structure of the ear, both external and internal. We divide the ear into three major parts, three major regions. There's the external ear, the middle ear, and the internal or the inner ear. And eventually we'll work our way into the inner ear where the receptors for equilibrium and hearing are. But let's work our way from the external towards the internal and talk first about the external ear. So the external ear, as we can see here, some components to it. Most of this is elastic cartilage with some adipose tissue, as you can see there. But the function of the external ear is to trap sound waves as they travel from the air, and you want to direct them, trap them from the air, and then direct them in towards the middle ear. So that's the structure of this auricle or the pinna, which is the external part that we visibly see. And that is that auricle or that pinna has a helix here, which is the rim of the auricle, and then the lobule down here, which is the inferior part, the earlobe. Tendons and ligaments attach the auricle to the head and keep it attached. And included in that external ear is this external acoustic meatus. Sometimes you'll see that is listed as the external auditory canal. But this is a curved tube that's about 1 inch in length, and it lies in the temporal bone and leads from the auricle to the tympanic membrane, or the eardrum. Now that tympanic membrane is the end of the external ear and the beginning of the middle ear. So that tympanic membrane of that eardrum is a thin, semi-transparent partition between those two parts of the ear, the external and the middle ear. And it's covered by an epidermis. It is composed of elastic fibers, collagen fibers, and it's rather flexible, right? It can vibrate in response to the presence of sound waves. Now, in the external auditory or acoustic meatus, there's also a few hairs called vibrissae that occur there, and some ceruminous glands which produce earwax, or also known as cerumen. Both wax and the ear in the external acoustic meatus both help to prevent dust or foreign objects like insects from entering the ear. Okay, helps alert you when an insect hits your external, or rather hits the vibrissae in your ear. That alerts the brain that, hey, something is in there and you should probably remove it. The earwax helps to trap foreign things from getting in too deep, because if they get in too deep, then they will eventually die and cause an infection. So cerumen and the vibrissae help to prevent things from getting in. Now the middle ear is this small air-filled, it's supposed to be air-filled cavity in the temporal bone that's covered in an epidermis or an epithelium. It's going to connect the external ear right here at the tympanic membrane to two small holes that enter into the inner ear. And so there's a round window down here, and there's an oval window right here where the stapes or this bone is attached. And that's how you get into the part of the ear which is fluid filled in the inner ear. But the external, or rather the middle ear, is air filled, and it has three of the smallest bones in the body in it. These bones are called auditory ossicles, and we've got the malleus, the incus, and the stapes in that order from the external towards the internal. Now the malleus attaches to the internal surface of the tympanum. The incus is the middle bone, and then the stapes is going to have its base actually attached to the oval window. That oval window leads directly into the inner ear. There's also a round window down here below the oval window. We'll talk about what that does in a few minutes, but it's also down here. That oval window, by the way, is not the oval, but the round window is sometimes called the secondary tympanic membrane. And we'll see why that is in a few minutes. Now, what I want to do is I want to look at it from a different perspective. Actually, I should have gone to this slide a minute ago, but there's a better view so we can see the middle ear and the inner ear a little better there. But I want to go to a different view of the middle ear. So we're looking here, notice at the top of the screen, where we have our view is looking from inner to the external environment. So here's the tympanic membrane. If we were to see through that tympanic membrane, we could see in the external environment. Okay, so that's our perspective. We're sort of sitting here in the inner ear and looking that direction. So we see the tympanum, we see the auditory ossicles here, but we also see some muscles, two major muscles here. One's called the tensor tympani muscle, and the other one's called the stapedius muscle. The tensor tympani is going to limit the movement and increase tension on that eardrum or on that tympanum. And it's going to do that to prevent damage to the inner ear, right? So when you're exposed to loud sounds, what you do is you contract that muscle, and that's going to pull back. It's going to create a lot of tension on the tympanum, which is going to prevent it from vibrating too much. The stapedius muscle does the same thing, but it's attached right here. It's attached to the stapes, right? And so when you contract it, it pulls the stapes away from the oval window, and it prevents it from slamming into it during loud sounds. So together, these two muscles make up what's known as the acoustic attenuation reflex. The acoustic attenuation reflex. This reflex is going to reduce the vibrations on the auditory ossicles to prevent overvibration and damage to that inner ear. All right. It's going to prevent you from damaging the inner ear where there's a lot of fluid and there's the receptors for hearing in there. If you put too much energy in that inner ear, it can damage those very sensitive cells. So the acoustic attenuation reflex is going to contract. It's going to be stimulated. It's going to make the stapedius muscle contract. It's going to make the tensor tympani contract, and that's going to prevent too much vibration in these auditory ossicles. Okay. By the way, the stapedius muscle, just to make note there in your notes, the stapedius muscle is innervated by the facial nerve, and the tensor tympani is innervated by cranial nerve number 5 or the trigeminal nerve. So make sure you remember that. Okay, so let me from this view, from this perspective, let me again say that one more time. As we will see in a few minutes, when we bring in sound energy into this external acoustic meatus, it's going to make the tympanic membrane vibrate. When that vibration takes place, it's going to then transmit that energy into these auditory ossicles, and they're going to move. And then you have this stapes attached to the oval window. Well, if I'm moving the stapes and I'm slamming it into that oval window, remember on the other side of that window, there's fluid. So you're going to create pressure waves in that inner ear. If I do that too hard, it creates really, really strong pressure waves, which can damage that inner ear. Now that's very important for hearing. We want to move the stapes against the oval window for hearing, but prolonged loud sounds is going to prevent that from happening. So everybody's been to a loud concert for an extended period of time, and so when you leave that show, you know that it's a little more difficult to hear for a while. That's because your acoustic attenuation reflex is still contracted. It takes a while for it to relax, and consequently it's hard for you to hear right after a loud show. Okay, let's stay here where we are. The other thing that I want to point out before we move into the inner ear is what occurs down here on the middle ear. We're still in the middle ear, and we see this auditory tube. Sometimes this is called the eustachian tube, and sometimes your book calls this the pharyngotympanic tube. So 3 names for this tube. But this is a tube that consists of cartilage, and it's going to connect the middle ear to the nasopharynx. Nasopharynx, remember, is on the in the nasal cavity, behind the nasal cavity in the pharynx region, so right above the oropharynx. Now, normally, it's going to be closed down here at the medial aspect, near the pharynx. But when you swallow or when you yawn, it's going to open up. The tube will open up, and it's going to let air from the atmosphere come in and equalize the pressure that's occurring in this middle ear. Remember that we said the middle ear is air-filled. And so if I don't allow any air to be moving in or out, In other words, down here, down here, right down here, if I have this area closed up, the medial aspect, no air can get in. So if I go, if I change my air pressure in the external environment, I go down in altitude or up in altitude, then the air pressure externally out here is going to increase. And that's going to cause a lot of tension on that membrane because it's not stabilized with the air pressure inside the middle ear. So the eustachian tube is primarily there to allow air pressure stabilization. When you yawn or you open your mouth real wide, that's going to open up that eustachian tube and allow air pressure to stabilize. Okay, finally, let's move into the inner ear. And specifically on the inner ear, we're going to start this off by saying that the inner ear is composed of a bony part and a membranous part. You also notice here the vestibular nerve going to the part of the inner ear called the vestibule. And you'll notice the cochlear nerve here going to the part of the inner ear called the cochlea. Together, these are going to fuse together or come together a little bit later, and they're called the vestibular cochlear nerve together. Now, the bony labyrinth, the bony labyrinth is what we primarily see right here. We're looking at a series of cavities in the temporal bone. And literally think of them to be cavities within that temporal bone. So we're sort of like looking at a mold. You put a mold into this cavity and then you pull that mold out. That's what it would look like. Okay, so it's just a cavity within the temporal bone. But that outer wall is called the bony labyrinth. And it's separated into three sections. So we have the semicircular canals up here. And those semicircular canals are going to have a membrane inside of them, I'll show you in a second, called the semicircular ducts. And at the base of each one of those, there's this little swollen area, and inside of that, there's a structure called the ampulla. I'll show you that on the next slide, I think. And then here, this is called the vestibule. This area here is the vestibule. It contains the membranous part inside of there is called the utricle and the saccule. I'll show you again in a minute. And then inside the cochlea, this snail-like structure here, inside there's a cochlear duct, which is the membranous part. So again, just to say that one more time, bony labyrinth is on the outside. It's the cavity in the temporal bone. And on the inside of that, there's a membrane. And that membrane is called the membranous labyrinth. Okay, so let's take a look at that a little bit more here. So we can see temporal bone here, and we can see what would appear to be the bony labyrinth on the outside right there, and then this blue material is all the membranous part. Okay, and so there's actually fluid, different types of fluid in each one of these areas. Before we look at those fluids, let me just remind us about these structures we just talked about. So up here in the semicircular canals, you'll notice that membrane in there is called the semicircular ducts. You'll also note that the ampulla is right here. Okay, that's the membranous part inside that swollen area at the base of those semicircular canals. So there's the ampulla. Right here is the saccule, and then there's the utricle. Okay, and then here is the cochlear duct. So all of that blue structure, that's membrane. And inside of that membrane is a fluid. And on the outside of that membrane, there's a fluid. So the fluid between the bony labyrinth and the membranous labyrinth, so that would be in this area right here, there's a fluid there called perilymph. Perilymph. On the inside of that membrane, on the inside of this blue membrane, if you go in there, there's a different fluid called endolymph. Okay, endolymph is inside the membrane. Perilymph is on the outside of the membrane. Now, what I want to just briefly do is to tell you kind of some functions, and then we're going to really focus in on hearing. So in terms of function, the semicircular canals, these are going to, or the semicircular ducts, they're going to contain the receptors. There's cells in there called hair cells that are going to be receptors for equilibrium. And so specifically, we've got the ampulla, as we mentioned earlier. The ampulla is going to contain receptors that respond to angular or rotational movements of the head. So spinning, when you spin, the ampulla there is able to detect that without you visually seeing that. In the vestibule, right here, we talked about the saccule and the utricle. That's the membranous part, the saccule and the utricle. And within there is a structure called the macula, which contains receptors for equilibrium also, but specifically these are going to respond to the pull of gravity. Position of the head, that's going to be picked up by the saccule and the utricle. And then in the area here called the cochlea, and the cochlear duct, that's where hair cells are going to be for the reception of hearing. And that's really where I want to focus in at this point. I want to go into the cochlea and see what's going on specifically within that cochlea so we can understand how hearing works. So what I want to do on this next slide is I want to take a minute to take a drawing. I'm going to take the cochlea and I'm going to uncoil it. I think it's easier if we stretch it out to see what's actually taking place. So I'm going to take this cochlea right there and we're going to uncoil it on this drawing. So here we have the oval window, the round window, and what I'm drawing here is the perilymph, or the bony labyrinth that contains the perilymph. So here is again my oval window, which is where the stapes attaches right there. And then here down below is the round window, a smaller hole that is contained by a membrane but no bone, no ossicles. So just inside the oval window there is a tube called the scala vestibuli, which contains the fluid we talked about earlier called perilymph. That eventually turns into the scala tympani, which leads to the round window. And all of this, again, is bony labyrinth that contains the perilymph. Internally, once you get into the membranous part, which is where we're going now, that's going to contain the endolymph. Now, within that cochlear duct, which is where we are here, there's a tectorial membrane. That membrane, the important part to note about that membrane is that it is stationary. So this is the cochlear duct. There's a membrane inside of there called the tectorial membrane that does not move. Okay, very important to realize that. Where the scala vestibuli and the tympani meets, called the helicotrema. And then the other part of the membranous labyrinth is going to be right here, which is called the basilar membrane. It's going to contain the spiral organ. We'll talk about that in a minute. The other part is the vestibular wall of the cochlear duct. Okay, those two membranes, those are membranes, they move. The tectorial membrane does not move, okay? But this part here called the membranous labyrinth, these components, they are movable. So when I move the perilymph and I move the endolymph, these membranes do move in response to that. So this is what the cochlea looks like when it's uncoiled. Now the way that your book depicts this, if we coil it back up and then take some sections through it, is like this. Okay, here's the way that the book shows it. So we have a cross section or a sort of a pie section here through the cochlea, as you can see to the left there. And then we see these coils. And so I can see when you take a cross section through something we just drew, then you can see here would be the scala vestibuli. There's the scala tympani. And then there is the cochlear duct, which contains the endolymph. So this purple color would be perilymph. And inside of the cochlear duct would be the endolymph. If we go a little bit tighter into this, and by the way, big picture here, nice big picture, is that you can actually see the cochlear nerve, right? Cochlear nerve, part of the vestibular cochlear nerve, but the cochlear branch of that comes here to receive information from cells that are down here in this organ called the spiral organ. So let's zoom in on one of these and take a look a little bit closer. Here we have one of those structures we just talked about. This is exactly what we did a cross-section of just a second ago when I uncoiled it. So this is looking directly on it. Here's the scala vestibuli, contains perilymph. Here's the scala tympani, contains perilymph. And here's the cochlear duct that contains endolymph. All right? What we said just a minute ago is that the vestibular membrane of the vestibular wall of the cochlear duct is movable. We talked about the basilar membrane being movable. But we said that this tectorial membrane right here is not movable. All right, it is not movable. What happens there is this basilar membrane contains an organ called the spiral organ. And that spiral organ contains the receptors for hearing. All right, so I'm going to go in a little bit tighter to the spiral organ. Here we're looking at the spiral organ. And here's the basilar membrane just for reference. Here's the tectorial membrane for reference. And notice that on the spiral organ, I have an inner hair cell, and then I have three rows of outer hair cells. These hair cells, specifically the inner hair cells, are the ones that are responsible for the sensation of hearing. The outer hair cells are going to adjust the tone of the sound. They're more motor, but the sensory part is the inner hair cell. Notice that all of these hair cells contain these little stereocilia, which are in direct contact with that tectorial membrane, which is stationary. It doesn't move. The basilar membrane does move, right? So if I move this basilar membrane at all, then it's going to cause the hair cells, which are in contact with that stationary tectorial membrane, to move. It's going to cause those stereocilia to move. And when that occurs, when you stimulate these cells by moving their stereocilia, that's going to communicate with these sensory neurons right here that lead to the cochlear nerve. So I want to leave it at that for a second. I want to step back, talk about something slightly different, and then come back to this and talk about how we interpret these different sounds. All right, so when we talk about sound waves, when people speak to you, their vocal cords are vibrating, and that causes the air in the atmosphere to vibrate. And the sound waves eventually hit your pinna, directed into your external auditory meatus, hits the tympanic membrane, and then your inner ear cells have to know what to do with it. And so when we talk about sound, there's two things we need to talk about. One is volume, and the other is pitch. All right. So volume, as this slide is indicating, volume is a measure of loudness. It's determined by wave amplitude. And amplitude is measured in decibels, which is the height of these waves right here. All right, so in blue, the blue line indicates loud sound because it has a high wave, it has a high amplitude. And then the green here is a smaller or softer sound because it's smaller in terms of its peak. Its waves are not as high. Now, when these sound waves hit your tympanic membrane, how does the brain know that it's a loud sound or a soft sound? Well, it all has to do with how hard you vibrate that membrane. If it's a high amplitude, a loud sound wave coming in, then it's going to cause that tympanic membrane to move a lot, and then it's going to cause the auditory ossicles to move a lot, and then it's going to increase the amount of pressure waves in the inner ear. So you're going to put that stapes up against the inner ear very hard, drastically, when you have a loud sound. And so there's going to be intense pressure waves. In contrast, when you have low amplitude or soft sounds, just the opposite is going to happen. The tympanic membrane will vibrate just a little bit, auditory ossicles vibrate just a little bit, and the stapes then moves just a little bit up against that oval window, which creates a little bit of pressure waves. And the brain can interpret that to be a soft sound because you're not stimulating as many hair cells that way. Okay, so that makes pretty much good sense. The loud and soft sounds have to do with decibels and have to do with the wave amplitude. Now, when we look at frequency, that's a completely different phenomenon here. When we talk about the pitch of a sound, high pitch, low pitch, that has to do with frequency, wave frequency. And so in red here, it shows a high frequency. This would be a high pitch sound. And the thing to note there is that the peaks between the waves are very close to one another, so they're short. Here we're looking side to side. So short waves versus the low frequency, the blue lines here, those are going to represent low pitch because they're long wavelengths. But notice on this particular graph that the height of these waves are exactly the same. So the volume in this case would be exactly the same between these two sound waves. The difference would be the high frequency is going to have a high pitch, the low frequency is going to have a low pitch. So the question is, how does the brain know? How does it interpret low pitch and high pitch if the amplitude, the wave height is exactly the same? How does the brain know that this is a high pitch or a low pitch? Well, it turns out that the wavelengths, if it's a short wavelength or a long wavelength, they travel into the inner ear at different places. So if you have sound energy coming in, I notice the stapes is here on the top, and as sound energy comes in at a high frequency, notice the wavelengths at the top here are very short. That's high frequency. Notice here in the middle, they're longer frequency waves. And notice at the bottom here, they're really long waves. Well, it turns out the high frequency waves don't travel that far into the inner ear, whereas low frequency waves do travel far into the inner ear. So they're going to stimulate, these sound waves are going to stimulate the basilar membrane, or the organ of Corti, or what your book calls the spiral organ. It's going to stimulate specific parts of the spiral organ at different places along the basilar membrane. So it turns out that if you stimulate this part of the basilar membrane close to the oval window, it's going to generate a high pitch. The brain will interpret that to be a high-pitched sound. The further you go to the right here in this uncoiled cochlea, the further you go to the right, the lower the pitch will be. The brain is going to interpret that to be a lower pitched sound. If you go too low in frequency, then we can't hear it. And the reason we can't hear it is because the sound goes right along the scala vestibuli. It never goes down into the basilar membrane to stimulate it, to make it vibrate. It just goes into the helicotrema and then down into the scala tympani and then right out the oval, or rather, right out the round window. So if you don't stimulate the basilar membrane at all, you can't hear it. For us to be able to hear something in terms of pitch, it has to hit the basilar membrane. That sound energy has to hit the basilar membrane at some point. So that's how we interpret sound energy in terms of volume and in terms of pitch. Big differences in how we actually do that. Okay, so let's summarize this with this particular slide. This is a pretty good slide that's going to summarize the physiology of hearing. So as sound energy comes in, sound waves come in, they vibrate that tympanic membrane at number 1. Second part they're indicating here is that the auditory ossicles are going to respond to that vibration. Right? The tympanic membrane is very flexible. It's going to move in response to sound energy hitting it. Auditory ossicles are attached to it, so they're going to move. We're going to transform sound energy into mechanical energy with those auditory ossicles. So then the stapes starts to move, and that's going to create pressure waves in the inner ear itself. All right? So number 3 says pressure waves created by the stapes pushing on the oval window move through the fluid in the scala vestibuli. All right, then at that point, if it's a very, very low frequency sound, as 4a here indicates, it says sound with frequencies below hearing travel through the helicotrema and do not excite hair cells. So it's going to come right out the scala tympani and right out the round window. So you won't ever detect it. But 4b says that sound in the hearing range, those waves go through the cochlear duct, they vibrate the basilar membrane, and then that stimulates these inner hair cells. So you'll notice here 4b, see how far it goes down here, and it stimulates only that part of the basilar membrane? That's where the hair cells are going to be stimulated, and that brain can then detect, oh, that part of the basilar membrane is being stimulated, therefore that part means this frequency. That's how the brain knows what frequency of sound is coming in to the inner ear. If it's really high frequency, remember it's going to stimulate the basilar membrane up here. Lower frequency gets pushed down towards the helicotrema. That's how we can detect that. Okay, great. So the final thing we need to talk about is the auditory pathway. So with all these special senses, we've been looking at pathways. Let's continue to do that here. And this is, I believe, our last slide. So this is the auditory pathway. So down here you can see where we just were in the basilar membrane. That's the spiral organ, which contains those hair cells. And there's the cochlear nerve as it comes, these neurons come out of that spiral organ. So they're first going to propagate along the cochlear neurons of the cochlear nerve, and they're going to synapse with the cochlear nucleus right there in the medulla. So you can see the cochlear nucleus in the medulla oblongata. It then moves up to the olivary nucleus. Okay, going to go to the olivary nucleus, and then to the inferior colliculus, and then to the middle geniculate nucleus, which is in the thalamus, and then to the auditory cortex, which is found in the lateral fissure just superior on the superior surface of that temporal lobe. That's where hearing is detected or interpreted. Now there's another thing that is not shown very well on this slide. I'm going to try to add it to it. Let's see. Okay, so this is what we talked about earlier, the acoustic attenuation reflex. And so that's going to actually originate in the olivary nucleus right there. So if you have really loud sounds, again, you're going to go along the trigeminal nerve. There's a trigeminal nucleus found here. This is going to then stimulate the tensor tympani muscle. It's going to make it contract. It's also going to go along the facial nerve. Here's the facial nucleus, and it's going to stimulate the stapedius muscle to contract. And both of those contracting muscles are going to create tension on the tympanum. They're going to pull the stapes away from the oval window, and you're going to reduce the amount of sound energy going in to the perilymph and ultimately into the endolymph. You're going to prevent the amount of damage that can be done by those hair cells, or to those hair cells, I should say. So this is the auditory pathway. We've seen pathways with each one of these special senses. Make sure you understand those. Make sure you can follow all those pathways. It's very, very important to understand the pathway of all these special senses.